Hi everyone, welcome back to the last video of the lecture. In this video, we will discuss more on the factors affecting the sleep-wake switch. Alright, so basically there are two key processes that involve in neurobiology of sleep. One is slowly changing influence, which is the homeostatic drive, and another one is the circadian process that depend on the light on particular nuclei, which is suprachiasmatic nuclei. Okay, let's have a look at the circadian rhythm first. So this circadian process linked to the internal clock, which is present in all cells of the body that we have. The circadian rhythm is closely linked to the light, and when light hits the retinal ganglion cell, it will translate via the retinal hypothalamic tract to the suprachiasmatic nucleus, or known as SCN. As I mentioned before, this SCN will signal to the pineal gland to turn off the melatonin production during the daylight. So exposure to the blue light during the day is important to suppress the melatonin secretions. Not only that, it is also important for keeping organism well-being, alertness, and cognitive performance. At night, no input from retinal hypothalamic tract to SCN, so the darkness will signal pineal gland to produce the melatonin. That's why melatonin acts on suprachiasmatic nucleus to reset the circadian rhythm. Okay. For the homeostatic process, on the other hand, is really need for pressure for sleep, and also this is a function of the time since you last had an adequate sleep. So if you haven't slept well, it is likely that the homeostatic process will kick in, forcing you to get an adequate sleep. Basal forebrain is considered the site of the homeostatic control of the sleep-wake cycle. Adenosine is the neuromodulator that play a key role in the homeostatic control of sleep. When adenosine increases to a level, it will act on cholinergic group in the basal forebrain and stimulate the hypothalamus VLPO to initiate the non-REM sleep. Okay, the sleep-wake cycle is analogous to a flip-flop electronic circuit. So this is a bistable device that will remain in one state until a trigger causes it switch to the other state. There are three main components in sleep-wake switch, which are the first one, hypocretin neurons, or known as the orexin, that is usually present in the hypothalamus. The second one, the local corollas, rough nuclei, and tuberomammillary nucleus that produce the monoaminergic nuclei. And also the third one, ventrolateral preoptic nuclei, or known as VLPO, that is responsible in secreting the GABAergic and melatonin hormone by the pineal gland. So we start with the arousal stage first. So orexin neurons, or we known as hypocretinergic, can be considered to be wakefulness neurons that stimulate or act to promote the activity of monoaminergic nuclei, such as in the rough nuclei, locus coeruleus, and TMN, to promote the wakefulness neurons, which at the same time, this monoaminergic nuclei will inhibit sleep promoting neurons in the VLPO. So in this slide, we will discuss on the reciprocal interaction of REM and non-REM sleep. So as you can see in this diagram, there is REM on and REM off. REM on represent the REM stages of sleep, and REM off can be either we are in the non-REM sleep stage or in the awake stage. So before we discuss more further on these reciprocal interactions, so I would like to highlight in the REM off. So the combination between the adenosine accumulation, especially in basal forebrain, as well as the VLPO, will inhibit the aminergic and hypocretinergic systems. So because of these actions, there will be initiation and maintenance of your non-REM sleep cycles. So non-REM sleep cycles is predominantly by the GABAergic neurons as well as some of the aminergic neurons. And meanwhile, the REM sleep is predominantly by the GABAergic, glutamatergic, as well as the cholinergic neurons. This hypocretinergic aminergic system will inhibit the cholinergic glutamatergic system, therefore inhibit the REM sleep. Meanwhile, the REM sleep can occur only when the VLPO release the GABAergic, galanin, as well as the melatonin concentrating hormone to inhibit the aminergic and hypocretinergic systems. So let's recap back what you have learned in this and previous video. So the chocolate box here represent the diencephalon, which is your hypothalamus and thalamus of the brain, which can be further divided into arousal state system, switch between sleep-wake cycles, and sleep homeostat in the basal forebrain and hypothalamus. Adenosine, okay, will inhibit the ventrolateral preoptic area from release the GABA or gamma aminobutyric acid, which cause wakefulness to us. However, prolonged arousal state may cause excessive of adenosine, which is part of the somnogen. Okay, somnogen is an agent that promotes sleep. Beside the adenosine as a somnogen, we also have the putative somnogens, including the cytokines, melatonin, oleomide, as well as the prostaglandin. Okay, back to here. This excessive adenosine will disinhibit the sleep-active GABA neurons of the VLPO, which then inhibit the wake-active histaminergic neurons of the tuberomammillary nucleus, as well as those of the pontine aminergic and cholinergic ascending arousal system, which from that will start the non-REM sleep. Okay, the same thing happened in the encephalic ascending arousal system, which the suprachiasmatic nucleus, or known as SCN. As I mentioned before, SCN are well known as a master circadian pacemaker that controlling the timing of sleep-wake cycles and coordinating with the circadian rhythm in the other brain areas and also the other tissues. As long as the photoreceptor being activated by the sunlight, the SCN will be activated and enhance the secretion of orexin in the lateral hypothalamus, and at the same time, suprachiasmatic nucleus signals to the pineal gland to turn off the melatonin production during the daylight. During the darkness, pineal gland will be activated to release the melatonin, or known as sleep hormone. Okay, this melatonin plays an integral role in falling asleep and staying asleep from awakening.